spatialization in India. So today we will discuss what is exactly the role of pharmaceutical biotechnology in the current era of modern drug discovery, how this pharmaceutical biotechnology is helping us for new biopharmaceutical therapeutic drug discoveries, and what are the different types of skill sets that may help researchers as well as academicians to design their projects, to apply for research grants, and to find out or to carry out the research in the field of drug discovery and drug development. So let's start with some basic fundamentals before going to the pharmaceutical biotechnology. Uh, if we want to differentiate between the basic research and applied research, so there is a major difference in the thinking process, or there is a major difference in the question we want to answer. The question we want to answer in the basic science is, why is this important? So this why is very important. So every time basic research or basic scientists ask the why to each and everything, to and so that they can reach to the roots of any particular scientific finding. While in case of applied research or applied science, we ask the question, how can I use this? And that is the most important thing, because if we discover something, or if you carry out some research work in your laboratory, that should be useful for people, society, so that the its applications can be exploited. And that is nothing but the how can I use this. So in basics research, basic science, it is completely driven, purely driven by curiosity and interest in a scientific question. The main motivation in the basic research is nothing but to expand our knowledge. And that's why most of the times basic science is theoretical, and the objective is nothing but to just get knowledge expansion. While in case of applied science, uh, we used to answer a specific question that has direct applications to the world, designed to solve the practical problems of the modern world, and that's why it is generally betterment for humans. So what both these researches are important, because basic research is actually the foundation of applied research. The applied research will be developed only on the findings in the basic research. So if we discuss some examples of basic research, then you can see here that how did the universe begin? This question may lead to the basic research findings. What are the protons, neutrons, electrons composed of? This may also the example of basic research. And what is the specific genetic code of the human? This is also the one of the example of basic research. So while in case of applied research, yes, the questions like how to improve the agriculture crop production is again the example of applied research, and we know the how this agriculture revolution has happened, and the green revolution has happened, and how it changed the world. Similarly, how to treat the cure specific disease. This is the area of interest of pharmacy people, where how can you contribute by doing research to treat particular diseases with the help of some new therapeutics or pharmaceutical drugs, or how to improve the energy efficiency of homes. So these kinds of researches are can be examples of the applied science. Yes, so but don't forget, the basic research lays down the foundation for the applied science. So recently, other than the basic research and applied research, you may heard about the term translational research. Now, the translational research becomes very important in case of pharmacy profession as well as medicine profession. Why? Because it is also known as translational medicine or translational science, where we are expecting the conversion of scientific discovery into health improvement. So ultimate aim of the any pharma profession or medicine profession is to provide the health improvement. If you are doing research, or if you are discovering something new, that should result into the health improvement. And this is the simple definition of translational research. So what is the translation? The translation of biomedical discovery across the spectrum. Now, what is that spectrum? That spectrum is very big. So that discovery or that research should get translated from the laboratory to the clinical trials, then from clinical trials to the clinical practice, and then from clinical practice to the population. So this is a complete zone or spectrum of the translation. Your research should get translated from the laboratory to the population. So that's why translation research becomes very important in case of drug discovery also. So translation research is multidirectional, multidisciplinary integration of basic research, patient-oriented research, population-based research. So all these aspects we have to cover in the translation research, and that's why it's a multidisciplinary. So we have to collaborate with different experts, experts from biotechnology, experts from microbiology, experts from life science departments, some clinician scientists, and always the pharma scientists also play a major role in the translation research. So here we are discussing translation research in case of translation medicine. So how can you justify the translation research for the betterment of pharmaceutical research? The the fact is that the translation research involves the application of knowledge gained through the basic research to studies that could support the development of new products. So ultimately, as a pharma person, we want to develop some new products, and that may be the pharmaceutical products or any health product which is better, which is supposed to be made for the betterment of people. And that's why whatever the knowledge you acquire during the basic research, that should get translated into the development of new products. So how you are going to translate that? So translate research in the field of medicine may involve using knowledge of the biology of disease. It is important to understand that you cannot discover a drug until and unless you are not or you are not known the cause of the disease, or you are not understanding the biology of a disease. So that's why disease biology is a very very basic research area which we neglect. Until and unless you don't have the idea of disease biology in detail, what is exactly happening in the disease at cellular and molecular level, you may not find the solution, or you may not find the drug for the particular disease. And that's why this knowledge of basic research or disease biology should be translated to the pharma scientists, so that we can have, or we will we will identify a very potential molecule. And that's why the next step is nothing but by understanding the disease biology, you can evaluate chemical compounds as a potential compound, so that you may find some of the desired compound in the from the bulk of the compounds. And the last step is nothing but the candidate drugs. Yes, if you screen the thousands of molecules, you may find some candidate drugs which are showing the desired effects you want against that particular disease, and you can convert that candidate drugs into the pharmaceutical drugs, and then only these drugs can reach to the clinical trials. So what it means? It means that from basic research to the clinical research, your translational research is acting as a bridge between basic research and clinical research. Similarly, from clinical research to the improved health, again translational research acting as a bridge. And that's why it is said that translation research is meant to bridge the gap between basic research and the clinic in order to facilitate the transition of knowledge and discovery into the therapeutics. Here we are discussing only therapeutics because we are supposed to be interested in the drug discovery. So what is the exactly translation research in simple words? Simple words, translation research is nothing but the bench to bedside concept. Now, what is the bench to bedside concept? Very simple. Whatever you are carrying out on the bench of your laboratory, that should reach to the bedside of the patient. As simple as that. So if you are doing research in your laboratory, if you are discovering something, if you are making some preparation formulations, or if you are discovering a new drug, that should reach to the bedside of the patient. And that is nothing but the bench to bedside concept. And that's why translation research becomes very important in nowadays. Okay, so unlike applied sciences, translation research is specifically designed to improve the health outcomes. And that's why we need team of experts, integrated team of experts, which can, or which who are actually focused on the translating the useful information from laboratory to doctors offices and the hospitals. Okay, so how can we contribute as a pharma scientist or pharma researchers in this field? So translation, so consider drug discovery and drug development is a translational research. You have to understand that the drug discovery procedure is not only the drug discovery procedure, it is a drug discovery and development. So here you need to understand that the drug discovery is the initial part of the pipeline, and drug development is the other part of the pipeline. So drug discovery is the first half of your research activity or research, right? And then second half may be the drug development. So ultimately you should start from the basic research, and then you are transferring this knowledge to the applied research with the help of translational approaches. So how the drug discovery cycle we can define? So drug discovery cycle starts from the disease biology. The first step of the pipeline is nothing but to understand the disease biology. It is important to understand the disease at molecular and cellular layer, why this is happening, what is happening at the cellular level, and how can I cure this disease. That is for nothing but the disease biology. Then second step is nothing but the target identification. This is the most important thing in case of pharmaceutical drug discovery, because most of the drugs fail into the market only because of wrong target identification, or the target identified is not as that potential as the required, and that's why they are unable to cure the disease completely. And that's why the second step it becomes very important, where you can identify the particular proteins which are playing a very major role in that particular disease. Third is, if third step is nothing but once you identify the target, then now you have to develop the assays to screen your molecules on that particular target. If your target is receptor, and if you want to antagonize that receptor, then you have to screen thousands of molecules to test that which molecule is capable to block that receptor or antagonize that receptor. And for that purpose you require some cellular assays or different types of assays to evaluate their effectiveness. Then if once you design the assays, now the next question is how to carry out these assays. And the fourth step is nothing but the screening of the molecules. So here we are screening thousands to millions of molecules at the initial stages, because we don't know which molecule is going to hit or which molecule becomes hit. And that's why we have to screen the number of molecules on the uh, target identified. And this screening step is again becomes very important, and here we use automation in pharmaceutical industries. Now out of these thousands to millions of molecules, once you identify few molecules or few hundreds of molecules which are showing desired effect on your target, these molecules are called as hit. Now these hits are, you have to convert them into the lead. That means from these 100 hit molecules which are showing the desired effects on your target, now you want to convert, or you want to identify 5 to 6 molecules that can become lead, or that have the maximum capacity to get converted into the pharmaceutical drug. And this can be done with the help of, and the next step is nothing but the lead optimization. Once you identify the lead, you can optimize the lead with the help of QSR and other techniques, so that you can increase their potency as well as efficacy. And then finally the 1 or 2 molecules may reach to the preclinical developments, and one molecule reach to the clinical development. And finally, after getting transfer from phase 1, phase 2, phase 3 clinical trials, and if you get the successful results in the clinical trials, your molecule may get released into the market, and you may get the patent here. Okay, so the goal of the drug discovery is to continue to accumulate the evidence, obviously, and then modify the disease outcomes in clinical population. And that's why the drug discovery and development, this complete pipeline, is a very good example of translational research. Okay, now what exactly this translational research offers you? Now, if you are interested in the research field, in particular pharmaceutical research field, whether you are academician or whether you are independent researchers or you are hardcore scientists, then only you have to understand the research gap. So many times in throughout the world, there are multiple grants or research projects have been sanctioned to the basic research. So like in India also, in national research centers also, most of the grants are given to the basic research. So you may find surprising that at national center, NCCS, NCL, there are crores of rupees grants are given to the basic research, just to study the different type of microorganisms or different types of animals, or sometimes different types of plants also. So yes, there are lots of research grants are available in the basic research field. There are lots of research grants available in the late stages, that is clinical trials, because here pharma industries are interested, and that's why they invest their money to carry out the clinical trial. And you may also get some funding here, but this is the gap where this is a funding gap. You may not get the fundings, you may not get the research projects. And this is the area we need to focus on as a pharma scientist or as a pharma researcher, because this is the area of drug discovery and development. And this is the classical example of a translational research where you can apply or design your projects, apply for research grants, and you have to convince the policymakers that they have to fund you, because this is the core area which need to be focused. Okay, now why this area need to be focused? Because in the current era, in the modern era, we are getting many types of biopharmaceutical drugs. So about more than 60% drugs have been replaced by the biomolecules or biopharmaceutical drugs. That means the future is all is our pharmaceutical industry future is nothing but the biopharmaceuticals. So in this case, it becomes important to understand the process of biopharmaceutical drug discovery and its impact on the practice of medicine. So every academician or researcher or the scientist in the pharma field should understand the importance of biopharmaceutical drug discovery and how it is going to impact the practice of medicine. And how can you understand that? Because if you once you get and understand detailed understanding in these processes, then you can contribute in this drug discovery and development field by your knowledge, with the help of some advanced technological tools. And one of that advanced technological tool is nothing but the biotechnological tools. So these biotechnological tools and techniques have been recently developed at very high end, and these biotechnological techniques actually resulted into the evolution of novel pharmaceutical drugs. So whatever the biopharmaceutical or biomolecule based drugs you are seeing in the market, most of the credit goes to the biotechnological techniques and tools we have discovered in last 2 or 3 decades. And that's why the pharmaceutical drug discovery or pharmaceutical industry is also changing or moving from pharmaceuticals to biopharmaceuticals. Okay, so this revolution, that is nothing but the biotechnological revolution, and it is slowly changing the pharmaceutical drugs also. And this revolution ultimately established the field of pharmaceutical biotechnology. And that's why the pharmaceutical biotechnology helps you to write the grants, write the research proposals, because in future, if you are writing the grant in or writing the projects, you have to include the high-end pharmaceutical biotechnology skill sets, or you will find answers of your scientific questions only when if you try to understand the cellular and molecular levels of the disease events. And that's why this is the solution for those people who are interested in the research activities in the field of drug discovery. Okay, so before directly jumping into the pharmaceutical biotechnology, let's discuss what exactly the developments happening in the biotechnology field. So if we classify, simple classification of biotechnology shows us the different kinds of biotechnology areas, just like green biotechnology, where biofuels, biomass, biopesticides, genetically modified crops, and bioremedications are these are the very highly grown areas where the biotechnology is playing important role. Similarly, red biotechnology, this is our area where biotechnology is helping to develop medicines, personalized medicine, genetic testing, diagnostics, health, telemedicine, gene therapy, tissue engineering. So most of the health related biotechnological applications are covered in the red biotechnology zone, where the blue biotechnology concentrates on the marine biotechnology, aquaculture, sustained solutions. Gold biotechnology is also helping pharma industry in the terms of bioinformatics, nanobiotechnology, where you can prepare different types of targeted delivery systems with the help of nanobiotechnology, genome sequencing, biometrics, biological computing, quantum computing, biosensors. White biotechnology actually includes the industrial biotechnology, enzymes as industrial catalysts, synthetic biology, and yellow biotechnology, where the food biotechnology, agri-biotechnology, nutrition, and nutraceuticals again, these are the industrial areas covered in the yellow biotech. So you can easily identify how this biotechnology is changing the world. Each and every factor or each and every area of industry, not only the industrial area, but on the different aspects of health, living aspects like biofuels, biomass, everything is dominated by the biotechnology. And that's why we always say that the future is only biotechnology. Now, the term biotechnology, you may feel like, oh, it's a new term. No, it's not a new term, because we have references in our ancient literature also. There were also examples of biotechnology, simple examples like in ancient era, the cheese preparation, or sometimes alcohol generation. All these biotechnology traditions were used in the ancient literature also, or ancient time also. So that's why we always used to say that biotechnology is nothing but the old wine in a new bottle. And that's why the terminology is different, but this science is not new. This science is completely old. But yes, we are developing this science for the betterment of people. Now, how this biotechnology is changing the human body? We have seen that how this biotechnology is changing the world, but what exactly happening, how this biotechnology is changing our understanding of the human body? So yes, due to the biotechnology, we better understand the proteomics now, because we know that the proteins are the actual functional players in your body. Each whatever functional in your body, that is protein, whether it is enzyme, whether it is coenzyme, whether it is hormone, everything is protein. So that means these are your functional players in the body, and this proteomics helps you to understand the role of different proteins in the different physiology, different proteins in the different pathophysiologies. Similarly, we can understand the different types of cells in our body, just like normal cells, disease cells. At the same time, we understand the stem cells in better, and that's why the stem cell area is now growing fast, and we are having the stem cell therapies also, which are again the very big achievement for the understanding of the human body. Similarly, nanomedicine discoveries are also possible due to the biotechnology, where we can identify or target specific drug delivery systems can be prepared with the help of biotechnology. Another area or another aspect of understanding the human body given by the biotechnology is nothing by the personalized medicine. Right now, we are giving one medicine for all, but biotechnology gives us or forces us to think about the pharmacogenetics and pharmacogenomics, where we are thinking about the personalized medicines. Why? Because we all are genetically different, and that's why we may require different medicines as per our genetic makeup, and that is the area where biotechnology is contributing significantly. Similarly, we can identify the gene variations, mutations, to understand the disease or to cure the disease at its root cause, and that is also given, that this gift is also given by the biotechnology. Synthetic biology is also the one of the future area, upcoming area, where the scientists are modifying or synthetically designing the cells as per our purposes, to convert these cells or to synthesize these cells, the desired substances, or to change the functions of these cells, so that we can use these cells for the betterment of our health. So you can understand how this biotechnology is completely changing the understanding of human body. So not only this, but biotechnology also revolutionized the pharmaceutical industries. How? Because nowadays, whatever we are listening is nothing but the biotechnology derived pharmaceuticals. I can give a simple example, like this in pandemic, we are having multiple vaccines in the market, and most of them are the biotechnology derived pharmaceuticals or biopharmaceuticals. So this is a very classical example where we have prepared new vaccines against the new virus, which was completely unknown, within a 1.5 year or 2 years of span. That is only because of the advancements in the biotechnology. Otherwise, you people also know the vaccine discovery may take 10 to 14 years to produce a new vaccine, but only because of biotechnological aspects and very high-end biotechnological era, we are able to produce some vaccines in this pandemic situation also. So, what is the future? The future of pharmaceutical belongs to the protein or biomolecules based therapeutics. Designing a stable or effective therapeutic proteins or biomolecules requires the knowledge of the structure of that molecules and how to stabilize that structure so that the function of that particular biomolecule should remain stable, and that function can be used for the therapeutic purposes. And this is the future of pharmaceutical industry. So yes, in the biopharmaceutical industry, the pharmaceutical biotechnology is playing crucial role, because it is a relatively new and growing field in which the principles of biotechnology are applied to the development of drugs or therapeutics. So majority of therapeutic drugs in the current market are biopharmaceuticals, such as antibodies, monoclonal antibodies, nucleic acid products, therapeutic proteins and peptides, new generation vaccines like COVID vaccines, etc. Such biopharmaceuticals are developed through several stages, similar to that of our old pharmaceutical drugs. So, as the way we develop the old pharmaceuticals, similar way here also the biopharmaceuticals are developed by stringent procedures, which includes understanding of the principles underlying the health and disease, then fundamental molecular mechanisms governing the function related to the biomolecules, synthesis and purification of the molecules, determining the shelf life of these biomolecules, biopharmaceutical. Again, this is the very channel, because now the pharmaceutical analysis becomes a very specialized area to analyze, to carry out the analysis in the analysis of the biopharmaceuticals. And that's why the bioanalysis is become very important than the chemical analysis. And that's why you have to develop the different skill set to maintain the quality control and quality assurance of these biopharmaceuticals. Okay, so similarly, the shelf life is established, is investigated. Similarly, the stability, toxicity, immunogenicity, all these aspects are also been tested for the new biopharmaceutical. Then, what type of drug delivery system may be suitable for that new biopharmaceutical, that has to be decided. And again, the new drug delivery systems are also developed. And lastly, the patenting and the clinical trials. So, yes, the biopharmaceutical drug also goes to the similar kind of drug development procedure through which the old pharmaceutical drugs go. So, what is the foundation behind the pharmaceutical biotechnology? The same foundation is simple. So, whatever the living, you have to use for your benefits. Now, the living things we have plants, microorganisms, animals, and fortunately, these plants, microorganisms, and animals produce low and high molecular weight compounds. And these low and high molecular weight compounds, whether they are biomolecules of their own nature, these can be converted into the therapeutics for the human consumption. And that is the simple basic for pharmaceutical biotechnology. So, yes, why this pharmaceutical biotechnology has flourished in the recent 2 decades? Because advances in the recombinant DNA technology, advances in the metabolic engineering, and advances in the bioprocess technology, these major 3 aspects are contributing in the development of the pharmaceutical biotechnology. So, the example is nothing but a large number of monoclonal antibodies and therapeutic proteins have been already FDA approved and available in the market for and they're contributing in curing the dreaded diseases for which we don't have the curable diseases right now. So I can give the example simple. We don't have the anti-cancer drugs or normal pharmaceutical anti-cancer drugs which are capable to cure the cancer. But right now we have some biopharmaceutical drugs or monoclonal antibodies as anti-cancer drugs, and these are curing the patients from stage 3 to stage 4. No doubt these therapies are very costly, we can't afford right now, but yes, but at least we have such kind of medicines, such kind of biopharmaceuticals in the market, which are based on the biotechnology derived pharmaceuticals, which are monoclonal antibodies, and they can cure you the cancer from stage 3 to stage 4. And that is the biggest achievement, and that's why in 2018 the Nobel Award was given to the such kind of discoveries. Okay, so yes, this is the basic reason the pharmaceutical biotechnology developed, and due to this rapid growth in the importance of the biopharmaceuticals and the techniques in the biotechnologies to modern medicine and life sciences, the field of pharmaceutical biotechnology has become an increasingly important. And that's why, as a pharma person or as a pharma researcher or as a pharma academician, you should be and you should understand the importance of this field. And uh, at the because you not only the in the research component, but also important component in the education of pharmacists, because you have to educate the students regarding this futuristic area, so that the students can appear or can make their career in this field only when if they have a sufficient knowledge about this field. And that's why the pharmaceutical scientists can contribute significantly. Okay, so broad aspects, can we identify the applications where pharmaceutical biotechnology plays significant role? Yes. So model system, cell biology, genetically engineered proteins, vaccine productions, just like I told, the new generation vaccines, virology understandings, disease biology to study the cancer research, drug discovery processes, experimental pharmacology, tissue replacements, biomolecules, genetic engineering, gene therapy, stem cell research, genomics, and toxicity testing. More major aspects of the research have been touched by the pharmaceutical biotechnology application, and that's why in case of pharmaceutical drug discovery also this becomes very important. Now let us jump to the conclusion why this pharmaceutical biotechnology is important and what are the different skill sets you may require if you are interested to contribute in this field, or as a pharma academician, a researcher or pharma expert, how you are going to contribute in this field. So in short, the techniques of biotechnology and the pharmaceutical biotechnology is becoming a driving force in modern drug discovery. So this is a very important statement. So whatever the techniques in biotechnology have been developed, and whatever the pharmaceutical biotechnology field has been established, this field and these techniques are actually becoming a driving force in modern drug discovery. So in the future, whole modern drug discovery will be dependent on these kinds of technologies, and that's why the biological scientist skill sets have been completely changed, and now we require pharmaceutical biotechnology skill sets, biological scientist skill set to understand the disease better, to develop the discover the new drugs. So these are the some important biotechnological skill sets, pharmaceutical biotechnology skill sets you may require if you want to become a good researcher in the field, or you want to apply for research projects or grants. You must include these skill sets in your experimentation. Then only you can carry out the research in depth. So what are these skill sets you may require? The cell culture, PCRs, flow cytometries, gel electrophoresis, Western blotting, confocal microscopy, ELISA, molecular cloning. I have just included the basic skill sets here you may require in this field, because there are lots of skill sets, but at least pharma scientists or pharma researchers should include these skill sets. You should understand these skill sets so that they can significantly contribute in the field of research. So let's discuss one by one. Cell cultures, yes. So cell culture is a modern era technique by pharmaceutical biotechnology toolset, where you can culture the animal cells or human cells in the laboratory. And this is a very big achievement for us, because the most important question in the research, or a problem in the research, is nothing but the in vivo in vitro correlation. So, and most of the times animal studies also we carried out, but animal studies have very few times very less correlation with the humans, and that's why it is important to carry out the research activities on the human cells. So this is the area where you can use the human cells in the experimentation, and you can test your molecules on the human cells, and whatever the findings you may have in these cell lines, that may have a more correlation to the human body, because these cells are already obtained from the humans. And that's why this animal cell culture has become a very important field in the field of pharmaceutical biotechnology, in particular in case of drug discovery also. So the process starts from the taking the tissue sample. You can take the tissue sample and trypsinize it, mechanically disaggregate the cells, or enzymatically disaggregate the cells to form a cell suspension. Now these isolated cells, you can convert them into the stable cell line by culturing them into the laboratory with the help of different cell culture apparatus, and different cell culture medium to provide the proper growth and nutrition. Then these cell lines get converted, can be transformed into the primary cell line, secondary cell line, tertiary cell line, and even infinite cell divisions. Now, how this cell culture has been revolutionized in last 2 decades. Again, these cell cultures started from the two-dimensional culture, and now the era has come where we have the very sophisticated techniques like 3D bioprinting, where we can form the organ in the laboratory also. You may find that the many foreign scientists have developed the ear or some kinds of brain parts in the laboratory by these high tissue culture techniques. So that's why it develops from the two-dimensional structure, where a simple cell surface flasks were used, and on plain surface cells were cultured and they grown in the flask. Then we slightly modified these two-dimensional cultures with the help of some floating membranes to provide some more physiological relevance to the cells. Then we apply, or we use the techniques like sandwich cultures. Then we use hydrogels, so that the cell and extracellular interactions can also be recorded in the laboratories. Then we go to the three-dimensional cell cultures, where spheroid cultures can be formed, because in our body the cells exist in 3 dimensions, not in the 2 dimension. That's why 3 dimensions always have more physiological relevance than the 2 dimensional. And yes, the 3D cultures, porous scaffolds have been used in the research, Then fibrous scaffolds are also being used in research. Now 3D printed models are also be available in high-end laboratories, and that's why this evolution of cell culture is actually contributing the field of drug discovery in a very significant manner. So let's understand the two-dimensional cell cultures in detail. These are the simplest form of cell cultures where cells are grown in a single layer. It's very important. It's single layer cell culture, and for that flask or petri dish are used. Uh, this technique is also called as anchorage dependent technique because the cells require substrate for attachment. They cannot directly attach to the flask surface. So these flask surfaces are treated when some substrate treatments are given, and with the help of that substrate, these cells get attached to the flask surfaces and they form the layer on the flask. The cells grow attached to the surface of the flask, then appropriate for the most of the types cells and the primary cultures, because this is the simplest form of cell culturing technique, and you can develop such kind of facility in your colleges also with the help of biotechnology expert. And again, it requires tissue culture treated vessels. Then it can be easily visually inspected through the inverted space microscope. No high-end microscopes are required. Inverted microscope can be sufficient efficient to show you the cells under it, and that's why the growth is again this techniques becomes very affordable. Then the growth is limited by the surface area. Again, this is the flask or particular plate culturing techniques. So the whatever the surface area of the flask is there, only that area is available for the cell cultures, and that's why as soon as the surface area get covered by the cells, you have to transfer that cells into the new flask, that is called a subculturing or passaging. So uh, these kinds of cell cultures require periodic passaging to transfer the cells into the new flask. The cells can be dissociated either enzymatically or mechanically. From transferring one flask to another flask, you can remove the cells simply by enzymatic means without hampering any properties of the cells. And yes, and that's why this two-dimensional cell culture becomes very popular in the field of research. Now, what are the advantages and disadvantages of the two-dimensional cell culture? Yes, the most important advantage you can see here that since it is a single layer culturing, so all the cells form a single layer covering on the surface of the cell culture flask. So whatever you are adding in the flask in the form of oxygen or nutrient, all these cells are getting uniformly exposed to the oxygen and nutrient, and that's why they receive uniform nutrients and oxygen. Similarly, if you treat these cells with a particular drug, then again the advantage is that all cells get uniformly exposed to the drug molecule. These are the biggest advantages in case of two-dimensional cell cultures. So yes, advantage is nothing but the easy to generate and maintain compared to the three-dimensional cell culture. Experimental versatility, you can modify your two-dimensional cell culture. You can see here, you can convert two-dimensional 2D culture by just giving some substrate treatments or adding some extracellular matrices components like laminin 1 to just produce the artificial atmosphere of extracellular matrix. You can form some 2.5D cultures here. Then also you can form some spheroids or spherical cell aggregates. You can modify your cell well plates with the help of some Boydin chambers, where you can study the behavior of cells, how the cells migrate, how cells invade. So these kinds of cell behaviors can be easily studied with the help of two-dimensional structure, and that's why these are the again advantages of the two-dimensional cultures, where you can modify your experiments. High reproducibility is another advantage, and obviously low cost compared to three-dimensional. Low cost is again the advantage of this two-dimensional technique. Now disadvantages, yes, obviously disadvantages, these are less biologically relevant. Why? Because inside our body, the most of the or almost all the cells are existing in the three-dimensional atmosphere, and here we are culturing the cells in two-dimensional atmosphere. That's why compared to the three-dimensional cell cultures, we have less biological relevance. Then lack of microenvironment, then unnatural adhesion forces are required because we are forcefully adding the cells to the surfaces, and obviously no cellular heterogeneity is achieved. So because our cells are surrounded by multiple different types of cells, and that is called as heterogeneity. So, but here we culture a specific types of cells to study their behavior. But yes, this problem can be solved by co-cultures or mixed cell cultures. Okay, so now let's jump to the three-dimensional cultures. Three-dimensional cell culture is a culture environment that allows cells to grow and interact with surrounding extracellular framework. So here, actually, we are providing the three-dimensional growth atmosphere to the cells, which are actually mimicking the uh, three-dimensional atmosphere inside our body. And that's why this is to improve the structure of cells and physiological equivalence in vitro. So we are designing the experiments in vitro in such a way that we are providing the cell three-dimensional atmosphere so that the cells can grow as they grow inside the body. And yes, and that's why three-dimensional cell culture refers to the culture of living cells inside micro-assembled devices. So for this purpose, we use different micro-assembled devices with 3D structure. And these devices actually mimic the tissue and organ specific microarchitecture. And that's why you are getting very, very close resemblance with the inside three-dimensional atmosphere. And that's why you three-dimensional cell culture, in three-dimensional cell culture, cells have better cell-to-cell contact, intercellular signaling networks, and we are allowing cells to differentiate into more complex structures. So these are the benefits of three-dimensional cell culturing. And here also, these three-dimensional cell culturing actually revolutionizing the drug discovery, drug screening procedures, because we are getting very, very close resembling results in these three-dimensional cultures. We can understand the cancer in three-dimensional. We can understand the growth of the particular organ in three-dimension, how these cells are behaving in three-dimensional, what is exactly happening in a particular disease in three-dimension. So all these benefits you are getting, and that's why for drug screening and validation, three-dimensional techniques have become more popular in pharmaceutical industries. Then similarly, you are getting more in vitro, in vivo correlation compared to the two-dimensional cell culture, and that's why the drug safety and efficacy assessment is also improved. So nowadays, 3D cell cultures and co-cultures receive more attention as they exhibit protein expression patterns and intracellular junctions that are similar to in vivo states. So whatever the cells produce inside your body, similar kind of proteins, similar kind of behavior you can achieve in your laboratory, and that is the biggest achievement in the three-dimensional cell cultures. And that's why 3D cell cultures have become increasing popular because of their more physiologically relevant and better representation in vivo tissue. So these are some techniques of three-dimensional cell cultures popularly used in the research field. The thin film insert substrates, cell aggregates at bottom of tip of drops, then liquid overlay, hanging drop, hydrogel embedding, spinner flask bioreactor, then scaffolds, different three-dimensional scaffolds, sponge-like structures are used where the cells can form their three-dimensional aggregates, and obviously the three-dimensional bioprinting, the cutting-edge technology has been developed now to form three-dimensional structures. Another cutting-edge technology beyond three-dimensional cell culture is nothing but the organ on chip. Yes, so the scientists have developed the small chips where you can develop the organ or representative organ on that particular small chip with the help of that organ specific cells. So here you can see that these are the scaffolds within the chambers. The scaffolds are provided within the chambers. This small chip contains small chambers, and that small chambers are supplied with a small scaffolds of three-dimensional scaffolds where the cells can live. Now, what is the advantage of this? For example, I can explain you this by giving the cancer example. So this is a normal cancer illustration, how the cancer grows. So these are the tumor cells. For example, breast cancer. So these are the tumor cells naturally develop in your body. Then in that case, these tumor cells are associated with basement membrane inside your body. They are in contact with the epithelial cells. They are in surrounding with the stromal cells. So this complete three-dimensional atmosphere is can be achieved inside the organ on chip in particular chambers. So same this in vivo arrangement of the disease can be achieved here in the chamber of the organ on chip apparatus. So this organ on chip here, these cancer cells have been put on the extracellular matrix membrane. The stromal cells are also cultured here, and obviously spheroid is formed in three-dimensional. That is the again advantage. And whatever the nutrition and other things required, these are given by these pipelines, small inputs where the picoliter or microliter of liquids have been supplied to this chip, and you can carry out the studies here. So, for example, you can develop the cancer, then you wait for the cancer to develop in the chamber, then you add the drug, and you find that whether that drug is reducing the cancer size or not. So, these kind of experiments, which are more correlate and organ specific way, you can carry out these organ on chip models. So is it imaginary? No, they have been developed and they are available in the research field. For example, liver on chip, skin on chip, brain on chip, eye on chip, gut, heart, lungs, pancreas. All these organs have been represented in the organ on chip concepts, and these organ on chips are right now available for research purposes. And that's why this is the organ on chip is a cutting edge interdisciplinary technology that can emulate the physiological environment and functionality of human organs on chip for disease modeling, drug testing, and precision medicine. So you can study disease modeling, drug testing, and precision medicine with the help of these kinds of high cutting edge technologies. So yes, these are some few examples where pancreatic cancer, glioblastoma cancer, then non-small lung cancer. Similarly, some preclinical trials, liver on chip have been already tested, developed. These chips have been developed and tested in the laboratories, and they found a very good research findings. They are very useful for research. Similarly, some first pass multi-organ chip have been also developed to study the pharmacokinetic values of nicotine. So you can understand that these organ chips are very, these are the platforms can be promising alternative to avoid the ethical issues related to the animal use, because for these three-dimensional purposes or organ emulation purposes only, we are using the animals for the studies, and that's why these three-dimensional organ on chip models can have a potential to replace the animal studies in future, and that's why these are becoming very popular in the field of research. So, next technique we are discussing is nothing but the skill set is nothing but the PCR. So, Carrie Bibulis, who discovered the PCR technique or method, uh, awarded Nobel Prize in 1993. So, this was not a simple technique; it is a Nobel Prize winning discovery. So, what is PCR? PCR is nothing but the polymerase chain reaction. And due to COVID pandemic, you might have seen the importance of PCR. So polymer chain reaction is a technique to make many copies of a popular or particular section of DNA. Section of DNA is nothing but a gene of interest. So your DNA may contains variety of genes. In what type of gene you are interested, you can isolate that gene, you can multiply that gene into the laboratory with the help of PCR, and that gene can be used for a different purposes. That purpose may be diagnostic, as well as that purpose may be drug discovery research also, or therapeutic purposes also. So what you require for PCR? PCR components are nothing but the DNA sample. Then you require primers, then you require nucleotides, then you require Taq polymerase enzymes, then buffers, and obviously the PCR machine and the PCR tubes. So what exactly the process of PCR? Yes, the DNA, double stranded DNA get denaturated in the first step at 95 degrees centigrade, so that the two strands get separated from each other. Then these two strands are get annealed with the primers in the second step, where at 55 degrees centigrade, these primers are the small, very specific sequences of nucleotides which get attached to that particular segment of the gene, so that only that gene grows, not other DNA part grows. So this gene only grows into the PCR machine and in the annealing step, and the last step is the extension. So this gene segment grows by using these nucleotides supplied externally, and these genes get replicated in the machine. And this is a very big achievement in the scientific discovery, because PCR actually revolutionized the understanding of genetics. Without PCR, it was not possible, because these genes are very tiny, very less inside. We can't even identify or detect them in their low concentration. We have to make millions of copies of a single gene, so that at least our detectors can detect their presence. So this is the limitation, and that's why the PCR plays very important role in these techniques. Now, how this PCR can be used for the research purposes? Now, in case of simple diagnosis purpose, the popularly you know that most of the COVID diagnostic tests are being carried out on the PCR. Now, let us take the example of the viral infection. So how the PCR actually helps you to identify whether you are COVID positive, or corona positive, or corona negative. So simple. If you are corona positive or COVID positive, then your body samples may contain extract of viral RNA or COVID RNA. That RNAs may be present in your samples, body samples, and that samples get these RNAs get extracted from the samples. Then these RNAs get converted into the complementary DNA with the help of enzyme that is called as reverse transcriptase enzyme, where RNAs get converted to DNA. This complementary DNA then form a DNA template for PCR, and then these for particular viral gene, COVID related gene, we add specific primers. These primers anneal to the specific region, and that RNA, that DNA get that gene get multiplied, form here, and then multiplied into the millions of copies. And these millions of copies give the signal in the form of fluorescence to the detector, and then detector identify that yes, that particular fluorescence is getting. That means this particular sample is positive in case of COVID, because this sample is derived from the RNA samples of the biological fluids. So this is the way diagnostic field is using the PCR. But now how research field can use the PCR? So there are two major variants popularly known as popularly in research field. One of the variant is the Rt Pcr, it is reverse transcription polymerase chain reaction. Here we actually convert or detect the RNA expression in the samples. So before treatment, after treatment, before disease, after disease, what are the different RNA expressions are changing? Such kind of studies can be easily carried out with the help of RT-PCR, where again you use the reverse transcriptase and you convert this RNA into the complementary DNA, and these complementary DNA transcripts are converted into the PCR and amplify them to identify the quantitative changes in the particular gene. For example, X gene is responsible, or X gene is supposed to be increased in the cancer. That X gene increased, getting increased or decreased. Yet this can be studied in the with the help of PCR. Similarly, another technique used very popular nowadays is nothing but the qPCR, quantitative PCR, or it's also synonymously known as a real-time PCR. This is also used to quantitatively measure the amplification of DNA using fluorescent dye. So in simple words, what is the difference between RT-PCR and qPCR? Difference is very simple. Both are very similar techniques, but in case of RT-PCR, we use or we detect the signal at the end of the PCR cycle. But while in case of qPCR, in you can detect the amplification of the DNA, how much DNA has been amplified in each cycle. That is the major difference. That means real time. At any real time, you can know, or you can identify that how much DNA has been amplified and how this DNA amplification goes. That's why this is the major difference between RT-PCR and qPCR. So these two techniques have been actually popularized in the research field. So many people think that, oh, that's what is the big deal? I can easily do the PCR. I can handle the PCR. I know the PCR. I can do experiments in the PCR. So what is difficult in that? So for those people, this is just summarization of different kinds of PCR techniques available, or different types of PCR available in the research field. So you may spend your entire life to understand these more than 36 different types of PCR modifications or versions for available for different kinds of research activities. So, and out of these 36, these are the only two we have discussed: real-time PCR and qPCR, sorry, RT-PCR, and maybe there are more different PCR modifications may come into the market in future. So yes, there are lots of FLP PCR, Hotstar PCR, In situ PCR, RT PCR, qPCR, Suicide PCR, Touchdown PCR. Different kinds of PCRs have been developed for different kinds of purposes. So what are the applications of PCR in particularly basic research and applied research? So this PCR helps you to understand the mutation screenings in drug discovery. I told you that the drug discovery is the basic research. Drug development is the applied research. So drug discovery, here also you can use the PCRs to understand what kinds of different genes are actually increasing or decreasing in a particular disease. And are these genes become a target for your drug? These kind of studies can be easily carried out with the help of PCR in drug discovery science, drug discovery research. Then classification of organism, genotyping, molecular archaeology, molecular epidemiology, molecular ecology, bioinformatics, genomic cloning, site-directed mutagenesis, gene expression studies. All these are the applications of PCR in the basic research. In case of applied research, you may find the genetic matching, detection of pathogens, prenatal diagnosis, DNA fingerprinting, gene therapy as the area of your PCR applications. The next skill set is the gel electrophoresis. Gel electrophoresis is a very popular technique in the molecular biology where we use the separation of biological macromolecules. We use this technique for separation of the biomolecules, generally proteins and nucleic acids. There are two major gel electrophoresis techniques: native page polyacrylamide gel electrophoresis and SDS page. So what is happening in the native gel electrophoresis? Native gel electrophoresis, we run the biomolecules in their native state. So we are not playing or we are not changing their natural structure. We are preserving the molecules in their natural form and we run them or we separate them as per their molecular mass. While in case of SDS page, we add chemical denaturant to remove the structure. So we unfold the natural structure of the proteins and we make them a linear protein so that we can separate them with the help of some gel electrophoresis principles. And that again depends upon their mobility speed. So these gel electrophoresis techniques are been easily used to identify, to separate the proteins, to separate the nucleic acids from each other, and you can use these techniques for the research. Next important technique which is based on the gel electrophoresis is nothing but the Western blot. Western blot is also popularly known as protein immunoblot, where you can study the expression of proteins in a particular disease or before disease, after disease. At the same time, you can also investigate the changes in the expression of proteins uh, before treatment of your drug and after treatment of your drug. So that's why the Western blot technique is again a very popular technique in the field of pharmaceutical research, where you can get the protein, isolate the proteins from the cell cultures or cells or samples or tissue samples. Then you can carry out the gel electrophoresis to separate these proteins as per their mass or molecular weight. Now you have to transfer these proteins to the membrane with the help of membrane transfer. And after transferring this onto the membrane, you have to block the membrane, and then you have to quantify these proteins with the help of primary antibody and secondary antibody, so that you may get the immunodetection and you can quantify that how much protein or which protein is present and how much it has been expressed in a particular disease or after particular treatment. And that's why the Western blot becomes very popular in drug discovery research skill set. And its applications are again with the help of Western blot you can study protein DNA interactions, protein-protein interactions, protein isoform detections, antibody characterization, epitope mapping, then subcellular localization of proteins. You can study post-translational modifications. So there are lots of applications related to the proteins Western blot can provide, and that's why it becomes a very important skill set in the field of research. Next skill set is the ELISA. So again, enzyme with the immunosorbent assay technique is again wet lab type of analytical biochemistry assay that uses a solid phase enzyme immunoassay to detect the presence of substance, usually an antigen. So here you can qualitatively and quantitatively, both purposes you can test your molecules here, or you can just detect the presence of peptides, proteins, antibodies, and hormones in your samples. And that's why ELISA is again become a very important skill set in the research, because it saves your time also and it can provide you the quantitative measurements also. So here also you can identify the samples, your samples, and here the major advantage of ELISA is nothing but you can test the soluble substances. So whatever the proteins, peptides, and antibodies and hormones they are present in the soluble form, that can also be detected with the help of ELISA that may not be detected in the Western blots. So here again, the reporter enzymes and appropriate substrates measurements are possible, and this technique is also very popular and very similar to the Western blot technique. What are the applications of ELISA kit? So it has a very broad scale of applications in food industry. Also, it has applications. Diagnostic industry, it has tremendous applications. And in case of pharma, we can study the pharmacokinetic principles and then anti-drug antibody reactions, host cell protein interactions in pharmaceutical research. Similarly, then agriculture research also has its applications, and biotechnology, particularly diagnostic purpose, it has tremendous applications. So this ELISA technique has three major principles where you can use a different kinds of approaches, where direct ELISA, sandwich ELISA, and competitive ELISA. These are the three techniques are popularly used for different kinds of research activities. Next skill set, important skill set or advanced skill set in the field of pharmaceutical biotechnology is flow cytometry. It is also known as fluorescence assisted cell sorting. So flow cytometry is another popular technique used in biotechnology. It is a laser or impedance based technique used for, this is completely made technique for the understanding of the cells. So whatever the cell kind of research need to be carried out in detail, these kinds of high end research can be carried out with the help of these skill set, flow cytometry, where you can study the cell counting, cell sorting, then biomarker detections, which are the different types of proteins are present on the particular cell, that can be studied. Then protein engineering can be studied, and that's why the flow cytometry allows the investigation of the expression of cell surface and intracellular molecules. So here, not you are not only just qualitatively identifying the presence of different kinds of proteins on the cells, but here you can quantify the expression of the proteins also, and that is the biggest advantage of this technique. Another advantage is that you can identify the different kinds of cells in the heterogeneous cell population. So in case of cancer tissue is always a heterogeneous population. So what kind of to understand the cancer at a cellular level, this is the very popular technique where you can understand or you can identify the different kinds of cells present in the particular cancer to know the heterogeneity of the cells. Similarly, you can understand the cell cycles of the cycles, apoptosis like processes in the flow cytometry, and that's why in the current era, flow cytometry skill set has become a gold standard for identifying the cell types with mixed population. So the principle is simple: whatever the fluorescent dye against particular interest of protein is added to the cell suspension, and that cell is transferred to the particular pipeline, and that pipeline is concentrated in a particular point where at one time only one cell can pass through that area, and during passing of that area, the laser light has been focused on that particular cell to study the properties of that cell. So different kinds of properties of the cells with the help of different antibodies, fluorescent antibodies targeting to the different kinds of proteins can be easily studied here. So counting of the cell is possible, cell sorting is possible to investigate the each and every type of cell protein on surface inside the cell. All this possible in the flow cytometry, and that's why this is a very unique cutting edge technology available right now. Applications, yes, it has very great applications for studying the cell activation status, cell cycle distribution analysis, cell division analysis, to study the apoptosis, to study the differentiation state of the cells, to study the cytokine secretion, activation of signaling pathways, calcium flux, level of intracellular reactive oxygen species, even the reconstitution studies and minimal residual diseases. So in case of drug discovery, yes, flow cytometry is helping in HIV therapeutics, screening of bioactive genes, biomarker discovery, small molecular drug discovery, drug action, drug safety assessments, drug efficacy, immunogenicity testing, pharmacokinetic analysis, and antimicrobial drug discovery. So this is a wide range of applications of this technology. So next skill set is nothing but the confocal microscope, fluorescent microscopy. It is an advanced version of the fluorescent microscopy, which is actually developed to understand the cell and in detail. We can understand, we can study the cell organelles also. So such high-end microscopy has been developed, and why it has been developed? Because the old fluorescence microscope, the entire specimen. So if you are testing a specific specimen or small tissue section, entire specimen is flooded evenly in a light. So the whole light is the light is given to the whole specimen, and that's why you are getting a very background emission of light, which actually interferes in the quantitative measurements of different types of expression of proteins. And that's why, to reduce this, the confocal microscope has been developed, where uh, to where we can reduce the unfocused background part. And here you the confocal microscope uses a point illumination at a particular point, very small area of the your specimen is exposed to the laser source, so that that small area of your specimen can be studied in details to understand their structural components at very high level. And that's why confocal microscope can be used to study the mechanism of actions of your drugs, investigate the mechanism of actions of drug, what exactly your drug is doing at cellular level, what exactly your drug is doing at cell organelle level. It can be easily identified with the help of these kinds of confocal technologies. So yes, again, just like flow cytometry, it has also wide range of applications, starting to study live cell imaging. Live cell imaging is the unique application of this confocal microscope. You can shoot the video of cell behavior or a live cell for 24 hours, 48 hours, what exactly that cell is doing in that 24 hours. That can be possible with the help of this microscopy. Then neuroanatomy studies, epitope tagging, neurophysiological studies, cancer research is the area dominated by the confocal microscopy, drug discovery, cell and tissue morphology studies, bioluminescent proteins, cell signaling pathways, resonance energy transfer, stem cell research is also dominated by the confocal microscopy, mechanism of action studies, membrane and iron probes, and DNA hybridization. So these are some beautiful images of confocal microscope. Now you can understand how detailing is possible with the help of confocal microscope. And this machine is very costly. It may cost from 80 lakh to 1.5 crore for a single microscope. Because of these kinds of potential of this, you can understand, or you can visualize the each and every part of the cell, in what is inside the cell, what is outside the cell. Not only this, but you can use the small fluorescent animal models under this micro. This is a zebrafish model where you can easily take the confocal microscope photographs. What is exactly happening inside the body of zebrafish? So you can tag some molecules inside with fluorescence, and you can track their movement of the molecules inside the zebrafish. So these are the very high-end applications of confocal microscopy. Next technique is cell-based assays. So cell-based assays are again, as I told you at the time of drug discovery cycle, yes, we require these cell-based assays for study the number of molecules to find out where their desired effects, to know the cytotoxicity assays, to know the desired effects of the molecules under investigation. The different types of cell-based assays are cytotoxicity assays, the popularly known as MTT assay. You might know that MTT assay is one of the example of cell-based assay. Then cell migration assays, cell invasion assays, kinase assays, protein assays, other metabolic assays. Why these cell assays are required? To study the mechanism of action of drug potency, receptor binding, receptor activation, cell signaling, drug internalization, subcellular localization. All these kinds of studies can be carried out with the help of cell-based assays. So yes, in case of drug discovery steps also, the high throughput screening technique is used to screen the thousands to millions of molecules on these cell-based assays to know the efficacy of the drug, to know the selectivity of that particular molecule under investigation, and to know the potency of the potential therapeutic candidate. Because we screen the thousands of drugs, then only 250 compounds may pass to the preclinical development, and then only 5 compounds may reach to the clinical trials. And all this requires the cell-based assays to screen these molecules for their desired effects. And these high throughput screening, the automatic robots have been designed to carry out these kind of assays on the automation level or bigger level to screen the activities of millions of molecules. And the next skill set is nothing but the molecular gene cloning. Again, this is nothing but the recombinant DNA technology, which actually revolutionized the biotechnological industries, where the target gene can be taken out from the particular source, and that gene can be incorporated into the vector to produce the recombinant DNA. Now, this recombinant DNA get introduced into the particular culturable living thing, for example, E. coli system or bacteria. Now, this bacteria system can be cultured in the laboratory to produce a multiple copies of genes. Now these genes can be used for different purposes, or that gene is responsible for production of a particular protein, and these proteins can also be used for different purposes. So here directly produced genes can be used for carrying out some new varieties, for example, gene for pest residence, resistance property for crops. Some genes can be transferred to the bacteria in the ocean, uh, for providing them a cleaning or toxic cleaning ability to toxic waste. We can use the proteins produced in these systems for the therapeutic purposes. For example, we have the proteins to dissolve the blood clots in case of heart attacks. We have some growth hormone like proteins which can be provided for those people who are not having a proper growth. So that's why this molecular cloning is also actually revolutionizing the biotechnology and pharmaceutical industry by producing or by helping in the different research activities like therapeutic product developments in just like vaccines, growth hormones, antibodies, vectors, recombinant protein, anti-cancer drugs. Genetically modified products or transgenic animals can be prepared with the help of these techniques. Fruits, GM vegetables, genetically modified crops, GM microbes, GM animals. Diagnosis is also the area where RDNA technology is playing important role. Gene therapy, CRISPR technology, which got the Nobel Prize last year. The monitoring devices, therapeutic strategies, energy applications, biohydrogen, bioethanol, biomethanol, biobutanol. All this can be easily obtained with the help of RDNA technology. So let's summarize the growth of the biotechnology in pharmaceutical industry. So I just discussed the basic skill sets you may require for carrying out the research in the pharmaceutical biotechnology. Now these are the some top-notch pharmaceutical industries players, biggest players in the world: Roche, Novartis, IB, Johnson & Johnson, Bristol Myers Squibb, Merck, Sanofi, and Pfizer. And if you search them, all these have been entered into the pharmaceutical biotechnology. All they are developing the biopharmaceutical drugs. Now, what is the status in the India? Yes, in India also, biotechnology industry is raising from 51 US million dollars to the 63 in 2019. Then expected growth in the biotechnology industry is 85 US million dollars in 2023, and by 2025 we are expecting 150 million US dollars valuation of biotechnology industry in India. So, what are the different sectors in India are growing with the biotechnology? Herbal tissue culture, micropropagation, then food nutrition, biofertilizers, diagnostic and biomedical devices, fermentation industries, and molecular biology industries have been rapidly developing in India with the help of these biotechnology advances. So, due to which, again, the clinical trial market in India is so rapidly rising, and in by 2025, we are expecting that 3.2 US billion dollars market will be there in India for clinical trials. So, in last, these are the some challenges for biotech industry. We have the limited research and development funding. We have strict regulatory rules. Again, the pricing is the major issue because research itself costs very high price, and that's why the pricing of the end products are again very high. So they may not be right now affordable to the common people. Again, involvement of patients is risky to carry out the clinical trials of these biopharmaceuticals is again more risky than the simple drugs, and obviously maintaining the innovation also requires a very high end understandings. So these are some Nobel Prize winning discoveries where the pharmaceutical biotechnology field, which discoveries actually strengthen the development of pharmaceutical biology. 2020 Nobel Prize goes to the CRISPR technology, to the Emmanuel Charpentier and Jennifer Doudna. Similarly, the Nobel Prize given to the medicine for oxygen levels, how the cells adapt to the minimal oxygen levels. Then Nobel Prize goes to 2018 for the two proteins identified by these two scientists, James Allison and Tasuku Honjo. These two proteins are the actually responsible for the uh, uh, giving breaks against the cancer. Uh, uh, for the immunocells, and these proteins now we are coming with the pharma industry is coming with the uh, drugs against these proteins, so that we due to these drugs we are able we right now in a stage to cure the stage 4 cancer patients in few cases also, and that case goes to the such kind of discoveries. Autophagy is again the area where we understand the cells in detail, and we are using that knowledge in the development of technological skills. I hope this has been a good learning for you people, and you can apply or you can develop your own skill sets for your research interest. Thank you.
Thank you very much, sir. It was very nice session, and you really explore the area of drug research and the transnational research in the field of pharmaceutical sciences. With your permission, may we
Thank
take
you.
some questions, sir? Hello, can we take some question?
Yes, yes, sure, sure. If anybody has question, yes, we,
Yes, sir. So, how pharmaceutical biotechnology used in IVIVC?
sir, your voice is breaking. Can you please repeat the question?
How pharmaceutical biotechnology can be used in IVIVC correlation?
Okay, IV-IVC correlation. Okay, what's happening in vitro in vivo correlation? That because most of the models we are studying in the drug discovery are animal based, and obviously the animal based IV-IV correlation is always tricky, because sometimes if you may find good correlation, and sometimes you may not find the good correlation. And most of the drugs get failed to show the similar kind of pharmacological activities or mechanism of actions in humans if you got some kind of activities in animals also. And that's why it is important to understand the in vitro in vivo correlation at cellular level. And in that case, if you use the human cells for that, human cell is the ideal sample, because you have the maximum correlation with you. So most of the cell lines used in the pharmaceutical biotechnology, they are derived from the human sources, and that's why they have obviously more correlation. The next part I describe is that the development of the three-dimensional cell culture. So three-dimensional cell culture gives you the more physiological relevance with the human body, because this three-dimensional atmosphere you can generate in the laboratory to understand what exactly happening inside the body, what exactly happening in that particular tissue. And that's why pharmaceutical biotechnology helps you to understand or correlate IVIV more in case of particular research, so that you can find good mechanism of actions of your drug.
Okay, thank you, sir. So, next question uh, is organ chip approach only applicable for preclinical, or it can be applicable for clinical as well as preclinical evaluation? Drug on chip.
Sorry, sir. Please repeat the question.
Is drug on chip approach applicable only for preclinical, or it is applicable in clinical as well as preclinical evaluation?
No, no, no. It may be applicable only for preclinical, because we don't have options for clinical trials. Because clinical trial we have to conduct, whether it is biopharmaceutical drug or whether it is a simple pharmaceutical drug. We have to go through the clinical trials. Yes, but it can give you the more confidence on your results, because in preclinical you may have findings related to the animals, but if you have the finding related with the human cells, and that too in the three-dimensional models, and that too in an organ on chip, so it gives you more confidence, and the failure rate in the clinical trial will be goes down. Because you have to understand that pharmaceutical industries invest crore of rupees or billions of dollars in the drug discovery cycle. And if any particular drug get fails at the stage of clinical trial, the pharmaceutical industry suffers a tremendous loss of money. And that's why this failure can be reduced by such kind of experimentation, such kind of techniques, so that your expenditure of pharmaceutical industries get reduced. And obviously if the expenses of the pharma industry get reduced, ultimately end product cost will also get reduced, and
Okay.
ultimately common people get benefited for that.
Okay. So one more question is there. What is DNA fragmentation or fingerprinting analysis? DNA fingerprinting analysis.
Yes. So can you repeat the question, please?
What is DNA fingerprinting analysis?
Okay, DNA fingerprinting is simple. Like everybody has its own DNA type, right? So just like thumb printing, we have all thumb designs. Similarly, our DNA patterns are also independent. So but these DNA these DNA fingerprints are also not matching with another people, and that's why these can be utilized for diagnosis purposes, or sometimes just to find out the sources of your origin also. So these kinds of DNA fingerprint techniques can be used just like a simple tool for know the DNA type of your own DNA type.
Okay, okay, thank you very much, sir. Sir, is it possible to share your contact details to the delegates if in case some delegates have question, they can directly contact
Sure,
you?
sure, sure. No problem. No problem. You
Okay,
can
sir,
share the mail ID also. No problem.
thank you, sir. Thank you. Uh, Sir, as a token of appreciation, I request you to accept this appreciation certificate on behalf of MIT's Institute of Pharmacy.
Aniket sir,
Thank you. Thank you. Yes, sir. Yes, sir. Good afternoon,
अनिकेत
sir.
सर गुड इव्हनिंग. मी तुमचे काही सेशन्सचं काही पार्ट ऐकला
Yes.
अँड आय मस्ट
Yes, sir.
फील प्राऊड अँड आय कॉंग्रॅच्युलेट यू, appreciate you because I have seen your journey
Thank you,
as
sir.
a
Thank you, sir.
lecturer to an expert in biotechnology, and the
Yes,
kind
sir.
of a
Yes.
command you have on the techniques
Yes.
which have been used, their applications, their principles, and your love for the biotechnology is
Yes,
been
sir. Yes, sir.
expressed when when you were talking about the different aspects and tools and skill aspects related to the biotechnology. So
Yes, sir.
let me
Yes, sir.
at the outset, let me first congratulate you,
Thank you,
appreciate
sir.
you,
Thank you,
because
sir.
I have a right to appreciate you. As I had said, I have seen
Yes,
your
sir. Yes,
journey
sir.
and feeling really proud about you, the kind of work you have done.
Thank you. Thank you. I
Huh?
really appreciate because you always give such a kind of opportunities where I can share my knowledge and thoughts. So it's really appreciated, sir.
Right, right. And looking for good association with you.
Sure, sure, sir.
Uh,
Sure.
Aniket sir, looking for good association. I know your future plan. So I see good opportunity for us to have a collaborative and a kind of a great work with a good outcome if we have a
Sure, sure, sir.
collaboration with you.
Definitely, definitely.
Huh? So we will be in touch
Yes,
with
sir.
you.
Definitely.
My team
Yes.
will be again in touch with you. Huh?
Sure, sure, sir. Sure. Sure,
Okay,
sir.
sir. Okay.
Thank you very much,
Thank
sir.
you. Thank you, sir. Thank
Thank
you.
you. Thank you, sir.
Thank you all delegates for your patient listening. Now, with prior permission of respected principal sir and the FDP coordinators, I declare that today's FDP session is concluded. All delegates tomorrow sharply at 10 a.m. you will join the YouTube link which will be shared by our team members. The link for fifth FDP will be and validatory functions will be posted on Telegram group and will be also posted on YouTube chat box. Thank you very much. The feedback
Okay.
link
Thank you.
Thank
and
you.
the certificate link will be posted in the chat box tomorrow during valedictory session. So with this note, I conclude today's session. Thank you very much.
Sure. Thank you,
Thank you.
sir.
Thank you,
Thank
sir.
you.
Thank you. Thank you.